My name is Dr. Raul Magadia. I'm an infectious disease specialist. I am one of the lead physicians at the COVID ward. I'm also the chairman of antimicrobial stewardship and infection control in both hospitals, Regional Medical Center Anniston at Stringfellow Memorial Hospital. So I'm here to talk about the basic science about vaccine in general, and in a little bit of detail on the technology behind both the Pfizer and Moderna vaccine called mRNA. So vaccine is a way of stopping infection. It's a great tool of stopping pandemic. What it does, when we give you vaccination, we're giving you maybe a part of that particular organism or a genetic version of it. In so doing, we are trying to entice your immune system, your body's immune system, to produce antibodies. Antibodies are highly specialized proteins that your body produces to help fight off infection. These are what we call memory cells. So once you get exposed to infection next go around, your body will remember that particular infection. So going to the mRNA vaccine, it has been studied several years now, especially in the epidemic that happened in Hong Kong, in Canada, called SARS, or Severe Acute Respiratory Syndrome, and in the Middle East called Middle East Respiratory Syndrome, or MERS. So the technology is there, but those two infections kind of miraculously, magically disappeared. So we didn't really have a way of finishing off the vaccine clinical trials back then. So what we did this time is we piggybacked on that technology, the old technology, and we used the knowledge that we've had with this new vaccine. There is a genetic material called mRNA, or messenger RNA. Messenger RNA is a specific protein, a genetic material. It goes to your cell, and the cell will makes copies of a protein called spike protein. Spike protein is a protein inside the coronavirus. So most coronavirus, regardless of mutation or difference in structure, all of them have the spike protein. So when the vaccine goes into you, your body encodes for the spike protein. So the mRNA does not go to the specific part of your cell called nucleus. So the question whether the mRNA will alter my DNA and all of a sudden I will develop some kind of cancer or some kind of autoimmune disease is probably far-fetched, because the mRNA does not go to the nucleus. So it has no way of altering your DNA, because DNA resides in the nucleus. So this mRNA technology only stops and does its thing in the cytoplasm, which is the cell body of the body of the cell. So that's one of the questions that asked of me all the time. One other question is, since the start of pandemic in March, and we get this vaccine approved last week, so it's span of several months. Typically, vaccine trials, they usually involve years, sometimes decades, where they're step skip. It goes through several phases. There's a phase called preclinical or pre-human. We call it in infectious disease word pre-human. So these are typically mice or some other smaller mammals, and they're given smaller amount of vaccine to see if it's effective. And if it is, it goes to a phase 1 clinical trial, wherein a bunch of volunteers, typically 18 to 25, mostly college kids, relatively healthy, maybe less than 100, and what we see, we give them several doses of vaccine, small, moderate to large doses of vaccine, to see if it's effective or safe, and we go to phase 2, which is a larger scale, maybe thousands, and after which we go to phase 3. Phase 3 involves maybe tens of thousands of clinical volunteers, and they have a specific endpoint, and the endpoint is prevention of symptomatic illness, prevention of disease, prevention of whatever they could think of. None of those steps from pre-human or preclinical to phase 1, to phase 2, to phase 3 clinical trials, none of those steps were skipped. The other question asked of me all the time is, what if I get infected? Should I get the vaccine still? The answer to that is yes. You might not be on the first batch. You might have to be pushed back further down the line. The scientific explanation to that is, if you get infected a month ago, you have protective antibody, or you're immune, so to speak, for at least 2.5 to 3 months, and after which your immunity wanes. So that means after 3 months, you're not protected anymore. And so you might get reinfected. The second scientific explanation to that is there's about 61, 62 mutations recorded so far. So if you get infected with one mutation or one strain, you're not protected against different strains. This vaccine will protect you against all the strains or even future strains because it encodes for a specific protein called spike protein, which is universally common in all coronavirus COVID-19 infection. The last question that's asked of me is, what is herd immunity? And it's a great question, especially with the vaccine coming in. Herd immunity is a way to stop a pandemic or stop infection by having certain population immunized, whether through vaccine or infection. So we've had herd immunity. It's not a new concept. We have, in the U.S. at least, we have herd immunity against mumps, measles, and polio. In measles and mumps, the herd immunity will be achieved if you have about 85% of people immunized or infected. With polio, it's about 90%. With COVID-19, the exact sweet spot is not really determined yet. Experts think that it's around 70 to 80%. What that means is 70% of the population, 320-something million Americans here, need to be vaccinated or immune for us to achieve herd immunity. And herd immunity affects two ways. One is it protects you and also protects your loved ones, especially those ones who cannot be vaccinated, newborn, pregnant women right now. So those are the ones we are protecting when we get herd immunity.